So here we're going to talk about toxicity associated with oxygen species, as well as cellular defenses. So again, the toxicity, we know that radical oxygen species are made through normal cellular processes, but they are neutralized as well through defense mechanisms. So the process towards oxidative stress, where you have too much reactive oxygen species and your host cell defenses cannot degrade them fast enough, then you start to get diseases associated with oxygen. So in general, oxygen radicals are going to react with anything inside of your cell, such as your proteins, your lipid membranes, your nucleic acids. Your peroxidation of your membranes is going to increase permeability of the cell. This can cause influxes of things like calcium. It can also cause mitochondrial damage. Mitochondria calcium influxes can overload. Remember that our TCA cycle is very sensitive to calcium, and calcium pushes TCA cycle forward. And if you continue to push TCA cycle forward, you're going to generate too much NADP, too many electrons. And oxygen loves electrons, so it's going to grab those electrons and make radical oxygen species. Transient ischemia is going to be associated with an increase in intercellular calcium. This is also going to activate your phospholipases, and it's also going to activate that mitochondria permeability transition, which then can lead to an influx and outflux of ions, damage to the mitochondria, and eventually apoptosis. So your radical oxygens can lead to protein, membrane, DNA damage. All of this is going to lead to cell swelling, increased permeability, influx of calcium, mitochondria damage, lipid damage, and eventually the death of the cell. Now, cysteine has sulfur molecules, and these are going to be a target for oxidative damage. DNA is going to be oxidized, and this leads to breakage in the DNA. Radicals have been associated with over 100 different diseases. Some of those we talked about in the previous lecture. And these radicals can also cause complications of other diseases. So the most susceptible targets for reactive oxygen species in our proteins are going to be proline, histine, arginine, and cysteine. Cysteine and methionine as well, are going to have those reactive sulfur species, and oxidation of this sulfur species is going to be reversible. So here's, for example, methionine, and it's going to have oxygen that's going to bind to the sulfur group and form a methionine sulfoxide. And then it's going to have another oxidation process which has methionine sulfonal. Now, when it happens on these residues, what happens is that you can get cross-linking of the proteins, or the proteins can fragment. Also, oftentimes they're targeted for degradation, so you lose the function of that protein. Iron is going to have the ability to bind to DNA, and this can facilitate the production of our hydroxy radical. Hydroxy radicals often are going to cause breaks in the DNA backbone or changes to the actual bases, so alterations of the changes, which can lead to damage of the DNA. Now, while all of this is happening, your cells are not defenseless. They do have enzymes that are very important in diffusing the radical oxygen species. So we have antioxidant defense enzymes, which include the superoxide dismutase, catalase, glutathione peroxidase, and glutathione reductase. In our diet, we take up vitamins that are coenzymes. Vitamin E is the major antioxidant. And then we have other vitamins that can be antioxidants as well, including our carotenoids, vitamin A, and flavonoids. Internally, we also make other things that can be antioxidants, including uric acid and melatonin. A process to protect against radical oxygen damage is compartmentalization of those, such as in our immune system, where they compartmentalize the radical oxygens into the lysosome. There is also a huge process to sequester metals such as iron, so you don't have the Fenton reaction occurring, for example. And then there are other repair mechanisms to replace damaged lipids, to replace damaged proteins, etc. So our enzymes that we have in our body are enzymes such as superoxide dismutase, or SOD. This is going to be very important against just normal oxidative stress. It's going to take that oxygen radical that grabbed an electron from the electron transport chain, for example, and convert it to hydrogen peroxide. So you have superoxide dismutase, and it forms hydrogen peroxide. For superoxide dismutase, you're going to find this in the cytosol, which is going to be associated with copper and zinc. In the mitochondria, it's associated with manganese. And then we also have extracellular. In the extracellular space, you also have superoxide dismutase that can handle superoxide radicals that can damage, such as extracellular membranes, collagen fibers, etc. And that will also be associated with copper and zinc. Another important enzyme that we have in our cells is catalase. Catalase can take that hydrogen peroxide and generate water and oxygen. It is going to be mostly in the peroxisomes that this occurs, and it's very important in very long chain fatty acids. So fatty acids that are over, so about 20 carbons, have to go through the peroxisome to be oxidized. And in this process, it always generates hydrogen peroxide. So catalase is very important in the peroxisome to neutralize the hydrogen peroxide into water and oxygen. Now there's also glutathione peroxidase. Glutathione is a tripeptide of glycine. We have glycine, cysteine, and glutamate. And so you're going to have hydrogen peroxide, for example, that was formed in the mitochondria from superoxide dismutase. And this hydrogen peroxide is still a reactive oxygen species, so it has to be neutralized as well. So it's going to be done through glutathione peroxidase, which is going to generate water, and it's also going to oxidize two sulfur groups. So you make an intermediate where you have two oxidized sulfur groups, glutathione disulfide. And then this glutathione disulfide will then be reduced through glutathione reductase, and this can be done in your cytosol with NADPH. It can also be done in your mitochondria with NAD. And so you're just basically going to reduce glutathione back, and then this can go back and work on another hydrogen peroxide, for example. Now, some non-enzymatic antioxidants are vitamin E, and this is the major vitamin antioxidants. So if you ever have a question about which of the following vitamins is an antioxidant, please pick vitamin E. This is going to be lipid soluble, so it's going to be able to protect your lipids. It basically can take the hit instead of your unsaturated fatty acids taking a hit. So it's going to be a terminator of free radical chain reaction, because remember, radicals occur in a chain reaction. And so the vitamin E will be that terminator instead of more damage to your membrane, for example. The sources of vitamin E are going to be vegetable oils, egg yolks, cereals. And here you have your alpha tocopherol. And then you're going to have your radical, lipid radical. And then so your tocopherol is going to pick up that radical and form a tocopherol radical. And then your tocopherol radical will pick up another radical, and then eventually it'll get reduced, and you'll form a quinone species. Now, while that's going on with vitamin E, your vitamin C is not an antioxidant, but your vitamin C can be a helper. So vitamin C donates electrons to vitamin E, and it can regenerate vitamin E. Vitamin C is going to be water soluble, so it has access to fluids, and it can get access to membranes through that, and vitamin C is also going to be a redox coenzyme for collagen. Another antioxidant is our carotenoids. So vitamin A, this is in fruits and vegetables. It's thought to have an impact at lowering cancer and eye sclerosis. It also is going to help protect the eye against macular degeneration of the retina. And this is in the lutein and xanthine components. Our flavonoids are antioxidants. They can chelate iron and copper. And so again, you can prevent the Fenton reaction from going on. So these are considered radical scavengers, and they donate electrons. Uric acid and melatonin can also be endogenous antioxidants. Purines are going to be degraded and form uric acid, and this is a major way to trap radicals that can then be excreted. It's important in upper airways to have this process. And again, uric acid is a form of getting these radicals out. Melatonin is naturally secreted by our pineal gland, and it's going to have the ability to pass through the blood-brain barrier. It's going to be an electron donor, and it donates a hydrogen, and this is to neutralize free radicals. It's going to react with both radical oxygen species as well as radical nitrogen oxygen species. And it does a suicide transformation, meaning that the radicals get neutralized. So radical species are known to have an influence on Parkinson's disease. So in Parkinson's disease, you have neuronal degradation. You have your dopamine levels because your dopaminergic neurons are degraded. The trigger for this is not known. There is monoamine oxidase, which is going to generate hydrogen peroxide. Superoxide dismutase in the mitochondria will also produce hydrogen peroxide. The mitochondria can get damaged, and it's thought that it leaks out iron. We have our iron enzymes in our mitochondria, and the free iron can lead to hydroxyl radical formation via that Fenton reaction. And then this is going to just facilitate radical chain reactions that damage lipids, proteins, DNA, and then you get neuronal degeneration and reduced dopamine release. So in general, oxygens, again, are going to contribute to cell death. They're going to extract electrons from normal biologic molecules. We have talked about superoxide, hydroxyl peroxide, etc., and they damage DNA, proteins, and lipids. They're going to lead to death of the cells, and our immune system, of course, is going to use this to destroy microorganisms because they have membranes similar to us, and so the process of damaging microorganisms is the same process that occurs with our cells. We also have defense mechanism, antioxidants, and we do compartmentalization to protect our own cells.